go to demands, So basically, I think we need to look at the hospitals, the teaching hospitals becoming referral hospitals. Just for that, I think that is where the problems lie. But in the private sector, these colleges here, the problem is that there are very few teachers, and you have people who have had no experience in teaching or learning to teach. One of the major things I think we should have is learning to teach, and none of them has a learning to teach. So
Vijay, I think for that you have to want to teach.
Sir, I think we should start the slides. Bhaskar is joining in 2 minutes.
Right. Okay.
Tufan sir?
Yes. Let us
Start the
start.
slides, please. We start the intro and he's just joining.
It is 8:10.
Today to 130th episode. We have a very interesting lecture today. How do you update yourself actually in this language of brain? So we will be talking about psychopharmacology. First part is today. Next slide, please. Handing over the session to Professor Dr. Tufan Pati sir. Sir is from Cuttack. He is our chairman of the program. Sir, over to you. Sir, you are muted. Please unmute.
Thank you, Alim. I welcome you to this episode of Thursday Musing with just 20 short of the 150th. And your blessings and your kind participation and your goodwill is the road. And thank you very much. Thanks, everyone. And without wasting time, I shall move on to introduce Dr. Alim Siddiqui and Amrit Patojoshi. Dr. Alim Siddiqui is professor of psychiatry and HOD Eras Lucknow Medical College. He is the treasurer of IPS. He has got all the money IPS has. And he is the vice president of IMA Lucknow. He is guest faculty at Pamit University. He is the honorary treasurer of IAPP UK chapter, editor-in-chief of Indian Association of Biological Psychiatry. And he has honorary fellowship IMA CGP 2021, 23 publications and presentations, 58 faculty lectures, 3 psychiatric book chapters, more than 100 newspaper articles. Dr. Amrit Patojoshi, neuropsychiatrist, professor and chief consultant neuropsychiatrist in High Tech Medical College, direct council member of IPS at present, editor of Odisha Journal of Psychiatry, chief coordinator of UNICEF, WHO, IPS Initiative and Telemedicine and Psychosocial Management. Welcome, Dr. Amrit. Both are vibrant moderators and steering heads of Thursday Musings. And let me have the pleasure and privilege to introduce our chairpersons. Next slide, please. Dr. Philip Jan. Thank you, sir, very much for accepting our invite to chair this session. He is a senior consultant psychiatrist from Cochin, and he had been in UAE in practice for over 42 years. Dr. Philip Jan graduated in medicine from Medical College Trivandrum and holds MD in psychiatry from NIMS. He is noted for pioneering the shift of psychiatry into mainstream medicine in Kerala, destigmatizing it in a general hospital setting. He also pioneered the introduction of child psychiatry services as a division of psychiatry, P-less Child Guidance Clinic in Kochi. Currently, Dr. Philip heads PJ's Neurocenter in Cochin, Kerala, a unique medical approach to the spectrum of clinical neurosciences with multi-specialty. Next slide, please. Dr. U. Gautam Das from Chennai is a professor of psychiatry, specialist in neurobehavioral medicine and expert in psychosocial intervention in disasters, educator and renowned psychiatrist in Chennai. He was conferred the Lifetime Achievement Award in 2015 by IMA. Dr. Gautam Das has been recognized by the government of Tamil Nadu for his services in the aftermath of the Super Tsunami in 2004. Dr. Gautam's handbook on community council trainers is recommended by the Disaster Risk Management Team of UNDP. Handbook of Community Council trainers is recommended for the Australian Disability and Development Consortium. Dr. Gautam's work has left a mark on psychosocial intervention in disasters and especially the involvement of women survivors. Dr. Gautam has been campaigning for awareness on learning disorders and was instrumental in introducing concessions for students with learning disability at Madras University, a provision that has benefited thousands of students. He is passionate about education of the girl child and has been facilitating process of education of girls from disadvantaged backgrounds ever since 2004. And avid environmentalist, he spearheaded the environment protection project of Rotary clubs in District 323 between 1991 and 1994, for which he was awarded the International Truth Wheel Prize for a Rotary Club of Salzburg for the best environment protection project in the world minus the Americans. Welcome, Dr. Gautam Das. And now I hand over the meeting to Dr. Philip Jain and Dr. Gautam Das. Please carry over. Next slide will be the introduction of Dr. Bhaskar Mukherjee. Can you introduce and then carry on? I am muting myself now.
Thank you very much, Professor Tofan. Dr. Bhaskar Mukherjee does not require any introduction to the psychiatrists of India. He says he's a student of psychiatry. All of us are, and many of us happen to be his students. As the medical discipline dealing with full working and dysfunction of the brain, molecular biology is where he feels most comfortable, and for him, psychiatry is molecular psychiatry. He loves to understand and evaluate the pathological basis of psychiatric symptoms, both macroscopically and microscopically, going from connectomics to genomics. His interest areas are treatment resistance in psychiatry. All of us actually take his help in the various groups, and transdiagnostic understanding of psychiatry, which is very important for us to merge into the medical discipline of psychiatry. And his life's goal is to make psychiatry regain. I don't know whether we have already gained the master spot in medicine, a spot reserved for the medical specialty dealing with the master organ of the body. As far as my outlook about Bhaskar's contributions to psychiatry in India, he has been actually part of this evolving psychiatry and the perspective of psychiatry in India into a medical mode. And I think psychiatry in India will always be remembered as Indian psychiatry before Bhaskar Mukherjee and Indian psychiatry after Bhaskar Mukherjee. Now, if you look at the contributions that Thursday musings have given me, there is a paradigm shift that we have seen. If you look at the past few years, it has been envisaged by the academic leadership in psychiatry. Professor Tofan, Aleem, Amrit, Bhaskar Mukherjee, I cannot forget Malay Dave, how much I've learned from him, and many others who have all come through in these webinars that we have been conducting for the past 130 episodes. Psychiatry, as far as Bhaskar has taught us, and many others of his class have taught us, psychiatry is to be perceived and practiced as the application of basic neurosciences to day-to-day human behavior, not as a storytelling psychiatry, not as a storytelling specialty that many of us were used to in our learning days. Thursday musings, as I said, 130 episodes ago, it started as the masterclass in therapeutics in psychiatry. And that's what we are taking on now. Bhaskar is now taking it on. He is walking his talk with his masterclass on psychopharmacology of brain, basics to applied. I think it will evolve. He is going to probably unleash it across to us as we go on in the next probably 1 or 2 episodes. Bhaskar today, I think, should be talking about in the ensuing sessions also, dealing with the genetic underpinnings, which is extremely important for him, as he has expressed in the introduction that I read out about him. Genetic underpinnings, epigenetic mechanisms, and the gene expression, which are all becoming extremely important for us to understand so that they become translational into our practice. the intraneuronal and the molecular mechanisms, interneuronal, the interneural networks mechanisms and the influences of psychotropics, neuroplasticity, as we would ultimately unravel, and the several related aspects of psychopharmacology in the brain, starting today with Bhaskar's lecture. I think Dr. Bhaskar has joined us. Has Dr. Bhaskar joined us?
Dr.
Yeah.
Gautam Das should give his preliminary remarks.
Yes.
Please,
Yeah.
please come.
So I was thinking, Dr. Bhaskar has told us he'll wind up in 45 to 50 minutes, and then we'll take a few questions as we get the time. Gautam, would you like to make an introduction? I was also hoping that Gautam would probably conclude today's session, talking about talking it as a summary of Bhaskar's presentation. I would understand much less of Bhaskar's presentation, I'm sure, than Gautam. So I think Gautam would make it clarify it for us in a summary. Thank you very much. Have a great evening.
Thank you, PJ. I think you are overestimating my capacities. But anyway, first of all, permit me to thank Dr. Halim, Amrit, Tufan, the Odisha IPS, the wonderful initiative that they have taken on, the Thursday musings. I remember how it started. But then it has been tripping along thanks to Alim and Amrit and team. And I congratulate them for the ongoing initiative, which I hope will transform teaching in psychiatry in India, if not across the world. Since we are already running late and Bhaskar is with us, I think I will hand over to Bhaskar to start the proceedings.
Thank you. Thank you, everyone. Sorry for being late. It seems I am always running on Bhaskar Standard Time rather than Indian Standard Time. Sorry. Actually, I am late day person. Means my day starts at around 11 to 11:30 a.m. and my day goes for practice. It is 10:50 to 11:30, and after that I sleep. I go return from work and do other things. So today it was a very tough day to conclude everything within 8 p.m. and so I'm very, very sorry for keeping you waiting. This episode is actually my proposed third episode of my series. The series started with basic concepts of psychiatry. Then we went on to describe various psychiatric symptoms in brain terms. And today and the next slot would be dedicated towards psychopharmacology, the real brain basics, and along with that, we would start giving examples, as well as we would start giving real life practice tips, which are science-based and which are translational in nature. So, without further delaying all of you, let's go to my screen.
You share your screen. We have 45 minutes.
Yes, and I have to do everything within 45 minutes. No problem.
Yep.
I am habituated in doing this kind of problems. This is Microsoft 360.
Yes.
And this is my screen. Okay. So this is molecular psychopharmacology of psychotropics. Now, first you have to understand that when we take molecular psychopharmacology, we take everything of neurotransmitter out of it. Neurotransmitter does not play a major part in most, if not all, psychotropics action. It's just the initial few seconds to few minutes change and nothing more. After that, it's a different scenario, and it's the real place of action of various psychotropics. I hope someday we would have a 7 or 8 lecture series on psychopharmacology of psychotropics from molecular and brain point of view. But in these 2 sessions, I would try to just show the overview, and I would show some pointers which would be good for practical understanding. With this, I am asking you some quiz. All of these are known to you, but still, these are some of the recap. We all know that in graft versus host disease, mycophenolate, mofetil, prednisone, rituximab, infliximab, all are effective. But we don't know that sertraline has a very good evidence for graft versus host disease treatment, and also and along with that, there are evidences for other antidepressants too. This is the actual patient-based study. Now, we all know that temozolomide, carmazine, bevacizumab, lormustine, doxorubicin are primary or adjunctive chemotherapy for glioblastoma. But none of us probably know that amitriptyline or trifluperazine or a lot of their congeners in psychiatry are as good as other neoplastic drug or more than that. This is a picture showing antipsychotics, TCAs, antiepileptics, and sedatives in glioblastoma multiforme. Similarly, actually, tamoxifen and endoxifen is anti-manic and pro-depressant. Penicillin is also the same. So they are not neurotransmitters. They are not neurotransmitter based. I am not going into epigenetic action because epigenetic action is one of the main action, but we have talked about epigenetics too much. I would just give the overview. Epigenetics means any action or any change in this to this pathway, I mean DNA to functional protein pathway. Any genetics or rather DNA derived control of this area, which is sometimes assisted by environment, is epigenetics. This is too much details of epigenetics, which we don't need now. If I remember correctly, I have talked about epigenetics in another national seminar just few weeks ago. So I am not trying to being repetitive. So the mechanism is this: DNA methyl transferase activity inhibitors. Here clozapine is one of these, along with amitriptyline, along with probably many of our well-known antidepressant. Histone deacetylase inhibitor, the most well-known is valproate, but many other anti-epileptic, a few antipsychotics fall in this. Histone methyl transferase inhibitor, many antidepressants actually work this pathway. Histone acetyl transferase inhibitor, many antipsychotic works here. Histone demethylase inhibitor, many antidepressant and second generation antipsychotic works here. Protein binding to acylated histone inhibitor. We are not sure how many psychotropics work here because this is something very relatively new. Similarly, protein binding to methylated histone inhibitor, again, this is something very new. So we don't know how many psychotropics work there, but more than a few work in this area. There is early evidence. This is a very old picture and often repeated picture in my presentation. Amitriptyline is DNA methyl transferase inhibitor. Valproic acid is actually DNA methyl transferase 3 initiator, or I would say augmenter. Escitalopram or haloperidol directly inhibit DNA methylation. Lithium, risperidone, olanzapine through microRNA inhibit methylation. In phosphorylation area, all antidepressants have role in DNA, direct DNA phosphorylation and direct DNA repair. Phosphorylation of histone is mediated by most of the well-known antipsychotics, including haloperidol and risperidone. Clozapine and olanzapine probably work here in case of histone methylation and increases the methylation of histone. MAO inhibitor through LSD1 actually also causes increased methylation. Fluoxetine, imipramine, amitriptyline, nortriptyline, and possibly fluvoxamine stopped histone modification. Amisulpiride through histone acetyl transferase or through direct mechanism causes histone acetylation. Similarly, all the activating antidepressants, including venlafaxine, desvenlafaxine, and other SNRI group, probably work through histone acetyl transferase to cause acetylation of histones. Fluoxetine inhibit histone acetylation, and histone deacetylase inhibitor like valproic acid and imipramine through that causes histone deacetylation. There are other various histone modifications in which psychotropics take an important part. This is microRNA pathway. I am not going into microRNA pathway, but their signature is all in all psychiatric symptoms, and through microRNA, we can say that many of the psychotropic action are mediated. For example, microRNA 146A, B5 and 425P and 2423P are markers of antidepressant response and regulate MAPK and WNT system genes. This is again a proof that various antidepressants work through microRNA pathway. These are some of the epigenetic effects of antipsychotic drug. Haloperidol in this induces DNA methylation in leukocyte, induces tissue and sex-specific changes in DNA methylation in rat models, induces DNA methylation in proliferative cancer cells. Clozapine increases trimethylation of H3K4, and similarly, all the psychiatric drugs work via these type of pathways. So this is another such table which shows effects of various antidepressant. I am not going into details of this because every year this number would increase and we would get more and more new epigenetic mechanism. For example, when this study was done, it was in 2018. This is a 2018 table, and contrast to that 2018, now we have more data about effect of lithium in DNA methylation, in peripheral blood, lymphocyte, and various other things. So every passing year would add few mechanisms to it. So I would not go into details of these mechanisms because the more we understand, the more mechanisms would be added. So we have seen that on chronic administration, all psychotropics changes gene expression as therapeutic action. So what are the results of this action? They changes the writers, readers, and erasers of cell, and which causes RNA modification, ultimately leading to conformational change of the DNA, translation of gene, cytoplasmic shuttling, functional on and off switches. RNA, it affects RNA stability, it affects conversion of non-coding to coding area, it causes secretion of new kinds of RNA, it causes various open reading frames to develop into new genes depending on environmental needs. There are lots of changes, and all of them are part of psychotropic action. What we don't know is which psychotropic is doing how many of these. So now we are going into more details of signal transduction pathway. I know this picture is very, very confusing, but this picture is a single picture through which we can talk about every action that psychotropics do inside cell. But that would be very, very complex. So we would divide into multiple small segments. For making anything antidepressant that has to act via TNF alpha receptor or TRKB receptor or vascular endothelial growth factor receptor or fibroblastic growth factor receptor, or possibly, this is a possible area. This is not really proven, possibly through serotonin transmission. All of these would cause activation of IP3, AKT, and mTOR pathway. Ultimately, it would change mTOR balance from mTOR1 to mTOR2, and that would lead to change in cellular autophagy and cellular apoptosis balance, and ultimately would lead to increased cell proliferation, increased cell cycle changes, increase in number of stem cells, increase in various forms of cell migration, and ultimately all of these would lead to antidepressant effect. This is one example that yes, fluoxetine works in mTOR signaling. That doesn't mean other molecules doesn't. mTOR signaling action is common in all psychotropics. Again, all the psychotropic work in different way in various WNT pathway proteins. But I am not going into details of it because this would be very confusing, and I promised that I would be more clinical than theoretical. So I would just mark few proteins to remember. SANK3 is one of the proteins that is widely used in animal model of autism, animal models of mania, animal models of psychosis. Similarly, frizzled is also one of those proteins which are heavily implicated in bipolar and psychosis, and all antipsychotic has some of the other action on these frizzled proteins. And GSK3B, the most known intracellular protein to psychiatry and psychiatrists. GSK3B action is there in every psychotrope. Lithium is the canonical one, but valproate works too. So is other antidepressants, antipsychotics, and various antiepileptics. Pimozide also inhibits WNT signaling, and that in a way help in hepatocellular carcinoma. This is a paper of WNT signaling and
Sorry to interrupt, Bhaskar, audio is breaking up.
I am not going into details of this because this presentation you are seeing. Hello. Okay. Now can you hear me properly?
Yeah.
Yeah, better.
Can you? Hello.
You're still
Yes,
breaking
you
up a
can.
little, Bhaskar.
Can you hear
If
me
you want,
properly now?
yeah. If you can, if you want, you can close the video so that we get a better
The
response.
thing is, this paper. Stop the video.
Yeah.
Okay. Now, am I better audible?
Yes, sir. Yes,
Yes.
Bhaskar. Please proceed.
Okay. The thing is, this paper was published in 2019. Once this paper was published and a variety of changes were found into this study between schizophrenia, so-called schizophrenia and so-called bipolar, a question arose that are the schizophrenics taken in this study truly representative of schizophrenia, and are the manic patients taken were truly representative of manic patients? And ultimately, due to this controversy, this study has been questioned for a long time. Hello,
Yes, Bhaskar, you're audible. It's
am
only
I
your,
audible?
yeah, you're audible, but you're,
Yes.
yeah, now
Okay.
it's.
Yes. So, although this study shows a lot of changes in WNT pathway between schizophrenia and bipolar, the inherent bias of who is schizophrenic and who is bipolar made this study more of a debated entity than a real scientific evidence. But instead of all the controversy, this study has shown that there are actually defects in various pathways in this type of patients, and when they are treated, the defects changes. So that means our drugs work in these areas of cell transmission too. Now, this is actually very simplified, or depending on your perspective, very complicated picture of jagged notch pathway. Another pathway where psychotropics works extensively, and due to these pathway, so-called neuroplasticity fast occurs. Notch pathway is active in autoimmunity, but also notch pathway is active in making neural stem cells from quiescent to active state. In brain, neural stem cells and neural progenitor cells reside in neural proliferative zone. This neural proliferative zone is situated in subventricular area and hippocampal area. That is why if we see patients of repetitive epilepsy or seizure, we would find that there is sclerosis in hippocampal area because the neuroproliferative zone cannot withstand so much electrical variance. Similarly, if we see any form of dementia, doesn't matter whether frontotemporal or vascular or Alzheimer dementia, the periventricular white matter hyperdensity is always there, because again, those neural proliferative zones are very sensitive towards any metabolic changes. Now, neural stem cells in these zones, they normally stay in suspended animation. Whenever there is need of new cell, notch pathway is the first pathway that makes these quiescent stent cells into active set. This is details of notch pathway and notch intercellular domain. I am not going into details of it because it would be too complicated for people who not yet have understood the parts of intercellular pathway. This is one paper which has already seen that these are the pathways through which antidepressant work. This is another paper which has already seen that hippocampal plasticity is regulated to notch 1. So what all of these would translate into? This ends our basic part. And now we would go into clinical applied part, which would be understandable for you. That all psychotropics are epigenetic drugs and drugs acting in cellular response and immune response, and their final actions changes gene expression profile. The neurotransmitter theories are misleading and only minor part of the picture. All psychiatric disorders listed in DSM and ICD are symptoms only. And the real disease producing these symptoms are transdiagnostic in nature and are genetic and epigenetic phenomena affecting whole body. Similarly, all psychotropics are transdiagnostic and multisystemic in their action. Should a tags of antidepressant, antipsychotic, mood stabilizer doesn't have any meaning. These are one of the most powerful human drugs which we till date have underutilized and misused because of our poor understanding of psychiatric disorder. With this, we are going into application of this knowledge into our day-to-day practice. We have understood that psychotropics are molecular pharmacology level drug. They act molecularly, and so they act in various real pathophysiology of all the symptoms of psychiatry. Standing there, can we get a real application of all these understanding? This would be topic of rest of this lecture and the next full lecture. In this lecture, I would show the intricate pathophysiology of migraine and how that migraine is generated by molecular pathology in brain and how different drugs can be used to change those pathology, and that's how we can get a better understanding of migraine treatment, and we would be able to increase our arsenal from just 1 or 2 drugs, maximum 3 drugs, to more than 100 drugs in case of migraine. So what is migraine? Migraine is fast and mildest expression of cerebral dysfunction and most closely associated with all pathophysiological phenomena of brain. Whatever happens in brain, be it some form of vascular insult, some form of autoimmune insult, some form of various toxic insult, some form of genetic disease, migraine would always be one of the most persistent and one of the most initial symptoms. Migraine or cephalalgia, whatever we call it. So this is our initial basic understanding that whenever there is increased homeostatic load in brain, means brain is subjected to functional pressure, either due to external environment change or internal environment change, that would translate into high information carrying load for brain. This would cause hyperactivity of brain areas handling the load. Now, this hyperactivity would cause coupling of electrical activity of those areas into protein production or translational activity of cells of that area. That would, so ultimately more cellular protein would be formed and more cellular protein would be recycled, because the more cell synthesizes protein, the more there is misfolded protein response, intrinsically or intrinsically disturbed protein response, resulting in formation of more and more disturbed protein production, and that would be metabolized and channeled in ubiquitin pathway. Ubiquitin result in endoplasmic reticulum stress, and that would cause cellular stress. Similarly, there would be increased mitochondrial oxidative phosphorylation, increased cellular metabolic demand, and this would result in redox stress and cellular stress. Again, this type of stress would be sensed by node-like receptor, which is a protein, which is a pattern recognition receptor in brain, and there would be resultant DMP pathway activation. This would lead to microglial activation by toll-like receptor. Microglia in turn would cause activation of astrocyte, and microglia would turn into M1 microglia, which is pro-inflammatory, and astrocyte would turn into A1 astrocyte, which is pro-inflammatory astrocyte. Now, pro-inflammatory astrocyte has negative impact on M1 microglia and would try to change the M1 microglia back into M0. This is called reciprocal activation and inhibition of microglia and astrocyte. And there would also be early activation of NLRP3 inflammasome in endothelial cell, which would result in neurovascular coupling. In neurovascular coupling, brain vessels dilate in response to increased metabolic demand of brain. Now, let's go into microglia astrocyte feedback loop. Microglia, whenever it get activated, it activates astrocyte, and activate astrocyte inhibit microglia. This reciprocal mechanism ensures that CNS neurovascular unit health and overall health and immune activation of immune repairs in brain stays in tandem. There are thousands of physiological and pathological processes that disrupts this reciprocal balance, and various type of cephalalgia, means headaches, are the first symptom of that. In physiology, this is the cells, glial cells. They have increased branching. They have various secretory activity. They have various metabolic activity. And whenever there is increased pathology, the branching would result in hypertrophy, rigid structure, and there is the microglia would turn into highly phagocytic and chemotactic. These are various signaling pathways that control astrocyte and microglia, reciprocal inhibition and activation. There is component system, there is norepinephrine system, there is purinergic system, there is various cytokines, there is various bacterial metabolite, and various other things. Now, all these control neurovascular unit of brain. So what is neurovascular unit in brain? Cerebral neurovascular units are formed by vessel associated microglia, astrocyte and their end plate, blood vessel muscle layer, pericyte and endothelial cells of blood vessel. They are innervated by various nerve fibers from trigeminal nerve, associated autonomic nerve bundle, and form small vessel Vasha Vasharam supplying wall of this large vessel itself. When there is increased metabolic demand in brain or brain excitation or local chemical changes, this blood vessel in neurovascular unit dilate, forming basis of neurovascular coupling. This is a simplified picture. This is junctional. These are junctional capillary associated microglia. These are wrapped capillary associated microglia. These are lined capillary associated microglia, and all of these are associated with astrocytes. Neuron innervate everything, including the microglia, astrocyte, and the smooth muscle cell of arteriole. The pericyte, these blues are pericyte. Pericyte wraps the capillaries in different area, which ultimately leads to this type of thing. Endothelium, pericyte, capillary associated microglia branching in between, and astrocyte end plate. So this is actual picture of our neurovascular unit, and this neurovascular unit secretes various vasoactive peptide and various inflammatory modulator during their activation. These are some of the inflammatory mediators. Unfortunately, this is an old picture of 2013-14 when calcitonin gene-related peptide was not that much important. Otherwise, along with everything, there would have been a large portion dedicated here to calcitonin gene-related peptide. This picture actually shows CGRP or calcitonin gene-related peptide here. This is another picture of the same neurovascular bundle from internal neuronal control and external neuronal control. External neuronal control comes from various autonomic ganglia, and internal neuronal control comes from locus coeruleus, raphe nucleus, basal forebrain, thalamus, various prefrontal areas, and the interneurons connects all these things and innervate vessels, astrocyte, microglia, and other things. If we do a schematic picture, then this would be something like this. Neurovascular unit is acted on by cerebral hyperexcitation due to anything. This causes the activation of neurovascular unit. Microglial activation due to anything would cause activation of neurovascular unit. Astrocyte activation due to anything would cause neurovascular unit. Peripheral autonomic activation due to anything would cause activation of neurovascular unit. Hypothalamic activation due to anything would cause activation of neurovascular unit. And this would lead to central immune system control, continued activation through neurovascular unit. Neurovascular unit would also cause peripheral autonomic activation. Neurovascular unit would also cause retrograde spreading vasodilation. It would also cause spreading cortical excitation wave. It would also cause retrograde autonomic ganglia activation. It would also cause endothelial inflammation and vasoactive peptide release, and it would also cause central autonomic activation. Now, each of these actions has certain ramifications, but let me show them. This is endothelial hyperpolarization proceeding, and there is retrograde hyperpolarization. Retrograde vasodilation and hyperpolarization. The gap junctions are increasing and other things. So this would result in spreading retrograde vasodilation would reach from capillary to penetrating artery to PL large artery, would cause gastrical pulsatile headache of migraine. Spreading cortical excitation followed by prolonged depression would cause migraine, aura, and autonomic hyperactivity, would result in hypervigilance, photophobia, phonophobia. Retrograde autonomic activation would cause increased autonomic ganglia downstream activation, photophobia, lacrimation, phonophobia, rhinorrhea, hyperalgesia of the skin, endothelial inflammation, and vasoactive peptide release, including release of all the vasoactive peptide plus CGRP, could cause local spread of vasoactive peptide like CGRP, VIP, substance P to different brain region and vessel, and they would directly irritate that. They would cause central and systemic inflammation and spread of pain. Central and peripheral autonomic stimulation would cause increased muscle tone in different muscles, including muscles of mastication, which are the most powerful fast twitch muscle in our body. So if these muscles are spasm, there will be bitemporal tenderness and pain and band-like sensation. So what we can understand from these things? It is a pan symptom of all dysfunction of brain. All drugs which use currently in migraine and all the drugs which would be better effective migraine are actually have specific effects in different step of pathophysiology of this symptom. Migraine needs long-term treatment with a rational combination of drug. Shuna headache syndromes are part of this whole process. And only thing we don't know now is which person would have which specific trigger and which specific manifestation. Now let's go back and just let's figure out action of all the drugs in migraine. This is the central picture. Cerebral hyperexcitation due to anything would cause neurovascular unit hyperexcitation and migraine. So any drug that reduces cerebral hyperexcitation would reduce migraine. Which are the canonical drug to suppress cerebral excitation? Number one always is anti-epileptic. That is why valproate is so widely used in migraine and other cephalalgia. But remember, cerebral hyperexcitation due to anything. So why only valproate? Give any other anti-epileptic. If the person is tolerating the anti-epileptic, then all the anti-epileptic is effective. Be it lacosamide, be it lamotrigine, be it topiramate, be it carbamazepine, be it phenytoin, be it even gabapentinoids. So see, this only one association expanded our armamentarium to include every anti-epileptic. Now, microglial activation due to anything. Microglial activation due to anything would cause neurovascular excitation. And neurovascular excitation would cause other features of cephalalgia. So if you stop microglial activation, then again migraine would stop. But microglial activations suppression would need time. So these group of drugs would need time to work. So what are those things? All so-called antidepressant suppresses microglial activation. So every antidepressant, not only our trusted amitriptyline or nortriptyline, you can give every antidepressant, including MAO, including SNRI. But yes, MAO and SNRI has another action, but I would talk about that later in the same slide. Astrocyte activation inhibition would also cause the same thing. Now, again, antidepressants does that. All antidepressants. Peripheral autonomic activation due to anything. Now, all the beta blocker comes here. Clonidine comes here. Our all alpha blockers come here. Hypothalamic activation due to anything. Now, what are the things that can cause hypothalamic suppression? Any understanding? Hypothalamic activation can be stopped by increasing satiety center of brain. And hypothalamic activation can occur due to any drive. That is why there is coital headache. Coital headache is one of the manifestation of hypothalamic activation. Whenever someone gets angry, there is headache because hunger is activation of hypothalamus. Whenever someone going through insomnia or forced awakening, there is hypothalamic activation. So satiety, on the other hand, would cause suppression of hypothalamic activation. Eating food would lead to satiety. Various other things would lead to satiety. And whenever there is satiety, there is less hypothalamic activation. So this is another way. Now, peripheral autonomic activation has a actually reciprocal action on neurovascular unit. And MAO inhibitors and SNRIs causes peripheral autonomic activation. So although they reduce microglial activation, they have a chance of increasing peripheral autonomic activation. That is why these two groups of drugs are better avoided initially when the headache phenomena is very active. Otherwise, initially headache might get more exacerbated and we might need to act various other things. So what other thing can we add? Cerebral hyperexcitation can also be stopped by calcium channel blocker all calcium channel blocker would cause less cerebral activation. So all calcium channel blocker can be used in migraine. Cerebral hyperexcitation can also be stopped by lithium. So all headache can have lithium as adjunctive treatment. Now, remember this. Potassium channel blocker, they actually can stop this pathway. And so all the potassium channel acting drugs, they also have a role in migraine or cephalalgia. Now, what are the other things? Autonomic ganglia activation. If we stop autonomic ganglia activation by ganglia block, we would stop the migraine. And there is various blocks that can be used, ganglia blocks that can be used in intractable migraine. Then, or endothelial inflammation and vasoactive peptide release. So what are the moieties that can be used as stopping endothelial inflammation? I don't know how many of you remember endothelin. Endothelin is one of the very powerful vasoconstrictor, and endothelin actually has specific antagonist. So endothelin antagonists can be given as a specific treatment of migraine. Vasoactive peptide release. CGRP got huge level of actually marketing, fame and other thing. But substance P inhibitors could also cause the same. Vasoactive intestinal peptide inhibitor drugs can also do the same. So this is how just knowing basic pathology and molecular pharmacology of psychotropics, we can revolutionize each of our treatment. So if you get a patient of migraine, we would start with first any autonomic blocker. Then we would add either calcium channel blocker or other acute acting drugs. Then we would start antidepressant with a caution that this drug would act after 4 to 6 weeks. And ultimately, this is the drug which would keep the migraine under control. And ultimately, we get 3 primary drugs: one antidepressant, one calcium channel blocker, one autonomic blocker for migraine. And later, if they do not work, we can get various other drugs which can be used as supplement. Supplement means second line, like other calcium channel blockers for this area. Or other centrally acting sympathologic, or other peripheral autonomic blockers like prazosin and other, or anti-epileptic or lithium or various immune mediator blocker. Who says that only CGRP blockers? I have already shown this picture. All these can, against all these factors, we got monoclonal antibody. And everything, specifically these, they have actually monoclonal antibody directed to it. So this is the benefit of knowing molecular pharmacology. Just by understanding pathology, you translate at least 1,000 new drugs for treatment of migraine, isn't it? With this, I am ending my presentation.
Thank you so much, Bhaskar. You can stop the screen share.
I have already stopped.
Thank you. Over to chairpersons for their opening remarks.
Thank you very much, Bhaskar, for that illuminating session. There's a lot of light thrown into the basics of our need to understand how psychotropics work and the various parameters from which the psychotropic can work. Two main things I think that come through today. One is that, that psychotropic drugs, he made a very salient remark today that they are epigenetic drugs. And therefore, the final actions are through changing gene expression. Am I right? Am I right, Bhaskar?
Yes. Yes.
And the psychiatric disorders, as we know them today, they are possibly a cluster of symptoms, and they do not truly represent a disease process which we can actually maneuver through using psychotropics. They are probably symptom clusters, and they actually represent genetic and epigenetic phenomena and manifestations. That too is right?
Yes.
That is one of the major takeaway that I have for today. So that, probably hoping that in your next translational presentation, I could use some of these to understand, as I was mentioning in the introduction, you have genetic mechanisms, the epigenetic mechanisms, the intraneuronal transduction mechanisms, you have the interneuronal mechanisms, and therefore the response to intervention, response to psychotropic intervention. The other major lesson that I learned today is about the neurovascular unit in the brain. And I don't want to go into with this, but then your explanation on how this neurovascular unit is impacted by various things that go on in the various subunits of the brain functioning. That is something that I need to actually look up, read, and then get familiar with. Thank you very much, Bhaskar, for that wonderful session. And I would now hand over to Gautam to take on from here and take the questions and manage the questions and probably summarize in the end. Thank you very much.
Yeah, thank you.
Please unmute. Yeah, thank you.
Yeah, thank you, PJ. So I think you have put it very succinctly. Now, I thought that I will just throw together a few slides to try to sum up what Bhaskar has come up with, and to try to steer, psychiatry in the right direction for the sake of the participants today. So will I be able to share my screen?
Yes, probably.
Yeah. Okay. So what I have understood today and what I have been struggling with over the last couple of years is that the more we understand about the brain, the more there is to understand. And therefore, the entire face of psychiatry is changing. There is a huge explosion of information in the last 10 years, which I'm trying to bend my head around. But all it boils down to is that we have to revisit the basics of cell physiology. The cell physiology that we learned in our time is totally inadequate, as also some of the understanding of what we called the silent cells in the nervous system. The astrocytes, the pericytes, the microglia, we did not understand in those days, and which Bhaskar so beautifully brought out when he talked about the neurovascular complex, which itself is a call to awaken ourselves into understanding the nervous system better, because otherwise we will be left far behind by our neurological cousins. So current classification is inadequate. I want to drive this home. I think this is something that we have to accept, that particular symptoms which involve neurobiological mechanisms are present across different diagnoses. Yeah. And so anxiety, depression, obsession, disordered thinking, mood changes all range from normal variation to pathological conditions. I think this is something that we have to accept before we can bend our head around the new psychiatry. As Bhaskar has pointed out, work on stressors has emphasized that environmental exposures become biologically embedded. So the problem starts right in the womb. And early diversity is associated with alterations in both body and brain. So psychiatry is not involved only with the brain, but with the entire system. It is a systemic disorder. So that is another fact that we have to bend our heads around. And it can be argued that DSM and ICD constructs are not sufficiently based in neuroscience. So DSM and ICD is a self-fulfilling prophecy of faith. Research findings demonstrate that there is considerable overlap of genetic architecture across DSM and ICD mental disorders. And if these current diagnostic constructs are not natural kinds, then any attempt to find specific biomarkers based on these diagnostic classifications and to develop targeted treatments based on them are doomed for failure. So we will be a dying specialty if we do not quickly pull up our socks. So psychiatry, I like to call it neurobehavioral medicine. Neurobehavioral medicine is a better term to describe psychiatry. And research has led to the understanding that particular domains of functioning involve specific neural circuits, which are in turn modulated by a range of molecular pathways, as Bhaskar has pointed out. And these molecular pathways appear to play a role across different mental disorders, and genomic methods have contributed to delineating causal and modifiable mechanisms underlying these conditions. So we have moved into genomics, which is the new science, which is beyond me, to be honest. So as Bhaskar has pointed out, all psychiatric disorders have been shown to have specific miRNA signatures. I have borrowed a slide from Bhaskar, who had been kind enough to send me all the slides beforehand when I just asked him to summarize what he was going to say. So all psychiatric disorders have been shown to have specific miRNA signatures, which could be the biological markers for these disorders. And psychotropic medication is shown to alter the specific miRNA signatures. Therefore, they work at the molecular level, at the cellular level. And psychotropic medication effect is brought about through signal transduction pathways, as Bhaskar has beautifully pointed out, which again, many of these things are a bit difficult for me to bend my head around, and reading and rereading, I don't think is going to help. So I'm trying to assimilate everything in a macro perspective, because it is impossible for my brain to go down to the micro perspective that Bhaskar's brain has gone down to. And all psychotropics act at various epigenetic levels and molecular levels, resulting in changing gene expression profiles. So I think this should be the takeaway today. So what we know now is that psychiatric disorders are manifestations of epigenetic alterations and molecular mechanisms. These alterations are modifiable or reversible by long-term treatment with a rational combination of drugs. And I'm going to say something blasphemous where Bhaskar is concerned, combination of drugs and psychotherapy, because psychotherapy, for those who favor psychotherapy, also works through epigenetic and molecular mechanisms, but it takes a much longer time to act and to bring about a lasting change in gene expression. We need psychotropics. What we don't know is which specific triggers manifest in the expression of epigenetic alterations in a person, and which trigger produces which specific manifestations. And that may take another 100 years. I don't know, Bhaskar should throw some light on this as to when he thinks this is possible to happen. So I open the floor to questions. Shall I pick up the questions from the chat box?
Yes, sir.
Yeah, we have a set of questions that have come. I think we should address that first in brief and then go on to the oral questions.
Yeah, sir, you can stop the slide share.
Yeah.
Gautam, would you pick up the questions so that they address the points which are relevant to his presentation?
Yeah. The first question is, do various psychotropics bring changes in the whole genome, or are they gene specific?
Whole genome probably is the right answer, because gene specific they are not. They work in multiple areas of DNA, many of which are non-genetic, and they also work in multiple gene areas. So it is whole DNA rather than a single specific gene. Fortunately or unfortunately, our drugs also work in another DNA, mitochondrial DNA. And I don't know which of them, because there are some I know which work here, but I don't know whether all of them or not. They influence the nuclear DNA and mitochondrial DNA interaction. That is why some of our drugs have limited role in mitochondrial disorders.
So I think that was quite comprehensive.
Very crisp.
Yeah. Savita ji has an interesting question. Does it mean that pathophysiology of migraine overlaps pathophysiology of mental disorders at the molecular.
Exactly. Or people would be very scared. But the thing is, pathophysiology of cephalalgia, or what the broad name of migraine, cephalalgia, is actually pathophysiology of mania, pathophysiology of anxiety, pathophysiology of all cerebral hyperexcitation in miniature form. That changes that we see in migraine, if we increase them in probably 100-fold or 1,000-fold, we would get the same changes in mania.
Thank you, Bhaskar.
So these are micromanics roaming around. These are micromanics roaming around.
There are thousands of people who have the vulnerability. What they don't know, or we don't know, is which trigger is for which person.
Okay.
There are persons, if I am not very wrong, I myself, a chronic migraine sufferer, has vulnerability to develop mania. But nobody knows, including myself, which are the specific physiological or pathological condition that would predispose me to have mania. That is the problem. If we have known that, then a lot of things could have been changed.
Which also brings me to a related construct, and that is of the so-called personality disorders.
Yes.
Which are probably the most vulnerable brains.
Yes.
Right.
They
I
are
think,
the.
Savitaji, I will come back to you. The one question is, would it be possible to develop diagnostic constructs based on these complex cellular pathways, and which would eventually pave the way for, what is that? Can't see. Okay. So can you at present, is it possible to develop a diagnostic construct based on these molecular mechanisms?
Yes, it is possible. It would be the future. But don't ask me to look into crystal ball and find out how long in future, because I have no idea. But ultimately, these names of schizophrenia, bipolar, these would be redundant. And the first crude diagnostic criteria would be disorders of motivation, disorders of executive function, and things like that. And then we would go deeper. The same way we have classified diabetes, the same way we have classified cancer, and the same way we are now revolutionized cancer. But probably, I don't know, I used to think 30 years. Now I have gone quite a bit realistic, and I am thinking now in terms of centuries, probably in next century.
Right. Bhaskar, I think you have opened a Pandora's box on migraine, because there is a rash of questions on migraine now.
Yes, I want them to ask about migraine, because I have just focused on migraine this session. In next session, it would be depression, psychosis, and mania, the big three.
Okay, there are a few questions on migraine, which I will club together. Why is there a unilateral headache, and why does it sometimes switch sides?
Because this specific neurovascular dysfunction, by its pathology, says that it should not occur throughout the brain every time. It should be a localized process for most of the time, and some of the time it would be generalized. So if it is localized, then being localized to one or the other cerebral hemisphere is the most normal course of action for these type of syndromes. That is why there is unilateral headache. But not all components are unilateral. The throbbing is unilateral, but if it is going to involve the other autonomic phenomena, then it is bilateral.
Questions on some of the drugs. For example, how do triptans work, and why are TCAs? Why have TCAs?
Triptans work. That is actually convention, nothing more. We human being loves to follow. Someone said TCAs work. Nobody asked why other drug doesn't work. If someone has asked before why other drugs doesn't work, then this question would not have arisen at all, and we would have been using every antidepressant, because I myself have used every antidepressant in migraine with good results. I seldom use TCA. Seldom. Let's say every 100 patient of migraine, I use maximum 2 patient, I would use TCA. Even less than that. This is answer of that question. And you asked one question before that. What was that?
Triptans.
Triptans is easy. Triptans have 3 mechanisms. Number 1, triptans work in such a way that they inhibit purinergic transmissions. Now, purinergic transmission inhibition means they inhibit the astrocyte, microglia, the interaction. This is number 1. Number 2, they are direct antagonists of CGRP related peptide, calcijorin related peptide. And number 3 is they are itself activator of endothelin secretion. Endothelin is something which is vasoconstrictor as well as some at a given dose anti-inflammatory and above a given dose pro-inflammatory. So by these 3 mechanisms, triptans work.
I think Savitaji, you can unmute yourself and ask your question.
Bhaskar, listening to you, the question is that how much of this molecular pathogenesis, pathophysiology that we have uncovered or understood is derived from the analysis or study of basic disease conditions, and how much of it is derived from understanding the effect and actions of psychotropic drugs? Or is it, the understanding of molecular pathology that we have understood, is it basically or largely derived from analysis and study of the disease conditions or the psychiatric disorders, or is it derived from the retrospectively from the analysis and understanding of this mode of action of psychotropics?
I would say 30. First 30% is understanding of normal physiology. That is the first thing. Then came the disease pathology. This is the second 30%. This probably would be more than 30%, around 40 to 50% would be this. And the rest is drugs, understanding of mechanism of drug. Because there are big loopholes in drugs. if you ask me what, let's say you give me a name of a specific drug and ask me what this drug does, I would give you, let's say 5, 10, 15 answers. But I know
It's
in
not
myself that I am probably missing more than 30 more.
right. So the drugs are much more complex, but the whole question and the fascination is about the discovery of these drugs, isn't it? I mean, how did we end up discovering
Yes.
these drugs and finding
Everything
them
is
out and
serendipity.
using them to their limited potential, whereas they have a vast potential, not only in psychiatry, but also in several medical conditions.
Yes.
So the discovery of these molecules,
Every medical field.
yeah, every medical field. So discovery of these molecules that means is the more a miracle than anything else.
Miracle.
Savita, there is
It
a
is
lot
miraculous.
there is a lot of serendipity attached to this, and
Yes,
there is
yes.
a lot of cascading understanding as we go on. For example, you look at lithium, how it came back. So there is a lot of serendipity that is associated with it. I have a set of concoction, 2 or 3 medications that I use for neurodevelopmental disorders, and they came back serendipity over the years, because in our country, when we use medications over a vast number of diagnoses, over a period of number of years or decades that you practice, you develop a pattern in your head, and you understand this works also for this, also for that, and that's how we become very often very subjective. As we see in the groups, we often become subjective. So basically, I think it's extremely important that we begin to look at these, working through the neural mechanisms, the psychopharmacology as it is related to the neural mechanisms and the molecular mechanisms. So basically, I think that's what would probably, in terms of time, may not be a century, but even before that, as the exponential evolution of technologies go, we might be able to understand as we go on. Maybe before we all would go, before Bhaskar goes, I think we would have much better answers than what we have today. Are we done for the day?
One last question for Bhaskar.
Yep.
Would you say that, given what you have just told us, that the so-called polypharmacy is the rule of the day?
It has always been the rule of the day. Problem is, we never acknowledge that.
True.
But I would add a single word before polypharmacy: rational polypharmacy. Because
great.
The shotgun approach doesn't work. The spray automatic rifle doesn't work. But if we make it rapid fire, it works. Because rapid fire means we are sending the actual thought out pathway. This is going to be the first one, second one, third one. Just like in cancer, no cancer is treated in single drug. There is no single drug treatment in cancer, because they know that in no cancer there is a single defect. But they mix and match the drugs based on their understanding of molecular pathology. Just like what I have given today on migraine, you would be able to use a rational polypharmacy in migraine if you consider this just. This is the way forward. Rational polypharmacy. Drugs chosen based on their action in molecular pathology of the whole thing and combining them. Then if they doesn't work, replacing each component with another thing that acts on a different component.
That is an excellent answer. Very impressive, Bhaskar. We had an excellent presentation. We really enjoyed. I thought it was going to be too hard for my brain, but I think I have a few takeaways, which is very encouraging. All of us do have the same feeling. the chat box has shown enormous number of congratulatory responses on your brilliant presentation. There is also a request for reading material.
My number is known to all.
Yes.
Just send me a message. I would send a book.
Right.
It's my responsibility.
Possibly for the newcomers and the youngsters, maybe you could give them your email ID or whatever in the next presentation.
No. Email ID is too boring for me. I am giving my personal mobile number.
You can do that. You can do
Bhaskar,
that.
please give it to us also.
Hey, तेरे पास है?
So with that, I think Gautam, it must be time for our exit to hand over to the remarks by chairpersons and the chairman. We had a wonderful session, and I think this is a take-home message so that we could bring this back to a translational approach in our practice with your next presentation. Thank you very much, everybody.
Yes.
Thank you very much, organizers. Thank you, Tofen. Thank you, Aleem and Amrit. This has been a great day. And Gautam, I'm really thankful to you. I always am encouraged by your, you say so many things out of the box, which leave a lot of thinking to do when we go home. Thank you very much. Thanks, everybody.
Thank you.
Thank you so much, sir. Thank you. Tufan sir, any comments from your side?
So, thank you very much. Thank you very much, Bhaskar, for your lucid presentation, your third appearance with us. And my takeaway is simplification of some complex processes plus confusion about some prefixed ideas. Thank you very much. And but what I look forward to, if we have proper big data and if you have proper application of AI into it, we can consolidate it into a form of regular pattern of approaching the patients, which will be easier for the clinicians. But may I mention here that my presumption is that NVN did not take up well, which is on that basis NVN did not take up well. But this is a thing that is needed in present day. We have AI, we have to generate big data, and we have to have the parameters. Then we can have a better, quicker result in the way Bhaskar thinks. That is my input, and I am enlightened as well as confused. Thank you, Bhaskar.
Thank you, sir. Amrit, Bhaskar has proven you are molecularly challenged, so it is your time to say thanks.
For the first time, I'll agree with Bhaskar. I am challenged and I have lost the challenge so many times, but thank you so much. We had a wonderful team, everything was going above my head. So I had to go to the top of the house to just catch something, but thank you so much. The chairpersons were better than the speaker, I would tell so at the end of the day. The first time we had the chairpersons overshadowing the star speaker, and that was great fun. And the best part was Aleem was also quiet this today. So I have a friend. So thank you so much. Catch you again next time. We had a great time. And catch you in time. Bhaskar, next time, catch you in time.
ये time problem है.
Molecular level में.
Time is a
नहीं सुनो, तुम सोचो 7:30 बजे म्यूजिंग्स है.
I will give you